Okay, welcome back from the long weekends. Hopefully you had a chance to recharge a little bit after that tough first week. I do want to ask, I have a student that is allowed to have a note taker. So if I just talk to me after class, if you'd be willing to be a volunteer note taker. So that would just require probably either taking a picture of your notes or sending your electronic notes to a student every week. That would be really helpful. So talk to me if you'd be willing to do that. Also, next week is our first exam already. So I kind of purposely put that early in the semester just to kind of get out ahead of that week where we have our lab exam. So it's not this Friday, but next Friday is your first exam. I have a little better idea now. I don't think we're going to get to a chapter that I thought we might get to. So the chapter 5 on eukaryotes, I changed the homework date for that to be due for the next exam. So if you haven't gotten into Top Hat yet, you need to do that for today, hopefully. I know a lot of you haven't started the homework yet. It's not a massive amount of homework, but it will do you more good if you are not doing it in a really rushed way. So kind of ideally do it as you go along. So get a start on that. Make sure there's not any problems getting into it or anything. And that homework is due next Friday, the day of the first exam. So I will post and give you an idea. I think that's going to include the chapter that we're going to start today. It's a pretty long chapter on cell structures. So I'm hoping to finish up the material for the exam by the end of lecture on Monday, and then hopefully not spill into Wednesday too much, hopefully not at all. So that's kind of where we're heading this week and next week. I continue to get questions on the grading of the homework. For those fill in the blank questions, you have to exactly match the capitalization, spelling, and plural or singular, or else it will auto grade it incorrectly. And I go in and regrade those, but if you can, if it makes sense for it to be plural, write it plural, and then that will just be a little bit less checking up on me. But I do have to go in by hand and do that, so it takes me a little time. So. Okay, so where we left off last time, we got to finish up microscopy. Yes, question.
For the homework, if you get one of those wrong, but it's actually right, notify you or
no, so you don't need to notify me. The way it's set up, I have to like scroll through all the students. Oh, here's somebody that's done it, and then I'll look at it. So I would say maybe look back at your homework grade after it's due, and if it looks, like, hey, wait, I thought I did a lot better than that, and maybe I missed something, then notify me after it's due. So yes, I'm kind of doing that little by little, and hopefully I will catch stuff that's just should be right. Yeah. Okay. All right, so finishing up our types of microscopy. So we presented these types of microscopy in the order of poorest resolution to best resolution, except I switched around the scanning and the transmission. So transmission gives us our electron microscopy, gives us the best resolution. That's the kind that's thinly sliced specimens. So the ultra microtome cuts those cells into whatever really, really thin sections. So what we're seeing is a cross section. This we can see the best resolution. And then the scanning microscopy, this is not cut thin. We're just seeing the outside of cells here. So this is more of a three-dimensional image. And I know that if you don't really know what you're looking at, it's a little bit harder to tell if it's 3D or if it's actually a cross section. Hopefully images like this, where you can see fine detail of a spore or some algae or whatever those examples there. I think these are all fairly obvious that they're the outside of what we're looking at. Again, not as good resolution as the transmission electron microscopy. When we prepare our specimens to view, it's still very time and very time. It's a real time sink because the specimens have to be coated in some kind of heavy metal, typically gold, and they're put on a little screen. And remember, they have to be viewed in a vacuum. So, you know, for many years we thought about getting an electron microscope here at Grand Valley, and sometimes I've heard of researchers at taking them a month or more to prepare their specimens to see under the electron microscope. So I ended up getting a confocal scope. Like I said, we have one over in Kinchi. So even, you know, if we're trying to, you know, diagnose a patient, if they have some kind of viral disease, we don't take specimens and look for that virus under the electron microscopy because it takes too much time and it's too expensive. Usually there's other things that we look for even to diagnose viruses. So remember, we can't see viruses with our light microscopy. We need that increased magnification, increased resolution that we get with electron microscopy. Okay, so just to wrap up here with a kind of quick overall introduction to staining. We talked about with light microscopy, you have to stain our cells most of the time in order to get good enough contrast to actually be able to see them. Otherwise, they look like their background. So almost all the time, you know, we're using bright field microscopy. We want to stain them. So just the basics here, you're going to get a little bit of this in lab, but if those are some of you maybe that are not in lab, we'll hit the basics here. Two kinds of stains. Some stains, the colored portion of that chemical stain has a negative charge, and we call those basic dyes. Okay, so the technical term is a chromatophore. We're just going to say that the dye has a net positive charge. I might have said that the wrong way. So the colored portion of the dye has a positive charge, and you know that unlike charges attract, so that positive charge is going to adhere to the negatively charged regions of cells. Primarily what we're interested in there is the cell wall, so the negatively charged cell wall. Okay, so this is primarily what we're going to use most of the time when we want to see bacterial cells, because it's just kind of easier. You stain the cell, now you can see it. So lots of examples of that, all the stains that you used for gram staining, for instance, what you'll get into gram staining next week. So this is going to be considered a positive stain when you stain the cells themselves. Okay. Conversely, if you use an acidic stain, that means the net charge of that colored portion is negative. All right, so that's going to be repelled by the negatively charged bacterial cell walls. Okay, every once in a while I hear a student say, well, the acidic dyes stain the positive charge cell walls. No. All bacterial cells have a slight negative charge. Okay, so when you're staining a cell, with a basic dye, you know, it's going to look something like that. When you're trying to see the same cell with an acidic dye, okay, we still get good contrast, but it's going to stain the background, and so the cell kind of pops out a little different way. All right, so we refer to this as negative staining. Okay, so again, positive staining, we stain the cell itself. Negative staining, we stain the background. And just kind of depends what we're trying to see as to which kind of stains that we would choose to use. A couple of other terms, if you're going to just do simple staining, that means that you would just add one dye to your cells. Whoops, use one dye. So you might add, say, a dye like crystal violet. That's what looks like what they used in this one right here. That enables you to see the shape of the cells, whether there's anything there or not, I guess. It doesn't give us a whole lot of other information, though. Most of the time and all the time that you're staining in lab, you're going to be using differential staining, and that means you're going to use 2 or more dyes. The reason that we would use more than one dye is because certain cells will take up one stain and certain cells will take up another stain. So then you can put your cells, decide what group they're in, or maybe that will enable you to see a particular structure better that you're trying to see. So for example, next page here, here's some examples of differential stains. And so primarily, this is kind of small, I can't even make that out. But the gram stain, that's a great example. We're using several different dyes. And they're listed here. You don't need to memorize these for me. You probably want to memorize them for your lab instructor, but you're using crystal violet, you're using iodine, and using safranin. And some cells are going to stain purple and some are going to stain pink. Some other examples, an acid-fast stain, an endospore stain, a capsule stain. Those are the ones that you're going to get a chance to do in lab starting next week if you're in lab. There is one other one that we don't attempt, and that's a flagella stain. It's supposed to be a lot more difficult to do, and so we don't bother trying it. I was going to go into a little bit more detail about the gram stain, but I think I'll leave that up to your lab instructor in just the interest of time. But I will talk about the importance of the gram stain. It's basically 2 basic types of cell walls that bacteria have. One big group we call gram positive, and one big group we'll call gram negative. Hopefully we'll get to those cell structures today. But it's the first question we ask when we're trying to identify an organism. Is it have the gram positive cell structure or the gram negative cell structure? Immense importance when you're trying to treat a bacterial infection. So you try to figure out what the infection is, and then that, depending on whether it's gram positive or gram negative, often will make a difference as to what kind of antibiotics are used to treat it. A lot of our antibiotics target the bacterial cell wall because that's a structure that we don't have in our bodies. So that's really common. So again, I'm not going to worry about the dyes that we use. The gram positive ones are going to stain purple, and the gram negative cell wall is going to stain pink. And we'll get to a little bit more why that is later. So, all right, any questions about microscopy before we move on? All right. So an enormously important chapter here, talking about cell structures. Okay, still really framework for a lot of stuff we're going to do the rest of this semester. So it's a pretty long chapter. And I've also changed the order quite a bit. The way the OpenStax presents a lot of this seems very jumbled up to me. So I have ordered it mostly, we're going to start on the outside of the cell and work our way in, spending a lot of time on the prokaryotic cell and then much less time on eukaryotic cells. So first, just we're going to review again the difference between prokaryotes and eukaryotes. It's one of those kind of big picture things that we want you to stick with you. We're going to also look again at cell shapes, cell arrangements, and then, like I said, we're going to move outside, move in from the outside in. All right, so again, little overview. Remember, prokaryotic cells, those are cells that are in the domains archaea and bacteria. Remember that Wos and Fox domains by ribosomal RNA sequencing. They are considered prokaryotes because they lack a membrane-bound nucleus. They also lack membrane-bound organelles, so things like the Golgi or lysosomes or endoplasmic reticulum, they do not have those. And remember, we said the size is usually around the order of 1 micrometer. Okay, that's kind of the typical size. You can see here the scanning electron micrograph of these bacilli-shaped organisms, about a micrometer. Compare that here with this protozoa. Looks like a paramecium there. You can see the units here, 40 micrometers. Right, so we compare the size there of the eukaryotes. I'll go ahead and write this in somewhere between 10 times to 100 times larger than prokaryotic cells. All right, and of course, eukaryotes, they have the membrane-bound nucleus, and they've got the organelles that have a membrane around them, like the lysosomes and the endoplasmic reticula, et cetera, et cetera. So examples of eukaryotic cells, we're going to touch on a little bit, probably not next week, the week after. Algae, fungi, plants, animals, protozoa, those are all examples of eukaryotic cells. From time to time, I will have a student that will get mixed up, when I ask the difference between prokaryotic and eukaryotic cells, they'll tell me the difference between a plant and animal cell. So that's sort of what's been drilled in you into maybe your biology classes. But remember, prokaryotic cells have cell walls. Some eukaryotes have cell walls. So algae, fungi, plants have cell walls. But we'll get into that a little bit more, the different parts as we go along. All right, this is a little bit, we talked about this already. That's one of the things that frustrates me about this OpenStax textbook is that they'll talk about the same thing like in multiple chapters, and then I can't remember, wait, did we talk about that already or didn't we? So we talked about little terminology with prokaryotic cell shapes. So we talked about the cocci and the bacilli and the vibrio. Those are all terms that you're going to want to know. It's a little nicer image, I think, here because it gives you some examples. We talked about the helical shapes, and maybe this is a good time to get a little bit more detail here. What's the difference between the sporillum and the spirochetes? The sporillum are a little bit more rigid, and they have external flagella. So we'll talk more about flagella here, hopefully today. And then the spirochetes, I've got another slide to show you some examples here. They are a little bit more flexible, and and they have internal flagella. So the flagella, as we see, are just kind of appendages that are used for locomotion. The spirochetes, their flagella is enclosed in a membrane, and it kind of is enclosed kind of down their body, their entire cell. Here's another example. You can't see the flagella really very well in these examples, but on the left here, these are the sporilla. You can kind of make out they're kind of tinted a little bit brown in this picture. It's kind of hard to see them, but the flagella are kind of off to one end in tufts. That's a scanning electron micrograph. It's just the outside of the cell. And the spirochetes on the right here, this is a little bit harder. I believe that's dark field microscopy, and that's a kind that we didn't talk about very much. I'd be hard pressed to know if that was dark field or if that was fluorescent microscopy just from looking at it, because they both have a really dark background. Let me just pull up real quickly, if this will let me do this animation just to see the motion. So remember, this is the sporillum. It always takes a little bit to find it here. Let's see if it's going to work. Maybe Not gonna find it very quickly. All right. Well, I'll have to let you peek at those on your own, those links if you're interested. You can kind of go down a rabbit hole looking for bacterial motion and things. But those are good examples of the spirilla motion, and the spirochete again is just a little bit kind of more flexible. It's kind of fun to watch. And then we also, it's very easy to categorize cells based on how those cells arrange themselves. So basically how they arrange themselves is based on whether do they stay attached after they divide. So, you know, one cell gets a little bit bigger and detaches into two cells. Do those stay attached together? And how many planes, different planes do they divide in? In other words, so if you have, you know, sometimes you might have cells that when you look at them under the microscope, you just see all single cells. If they tend to make pairs like this, well, they are probably just dividing in one plane and then maybe only stay stuck together for a short period of time. If you've got a group of 4 cells, we call those tetrads. Okay, so that's cells that are probably dividing this way, and it's kind of hard to draw this one. I don't have a 3D way to do it. So this way and this way. There's another fairly common cell arrangement that they don't mention here, the Sarsina. And these are kind of packets or cubed shaped packets. So instead of 4 cells, you're going to have 8 cells that stay stuck together. So it's dividing in an additional plane this way. Okay, and when you look at those under a microscope, it's kind of hard to tell when you have a 2D image whether there's 4 or 8 there, but sometimes you can tell. The streptococcus are the ones that make chains. That prefix strepto means chains. And then staphylococcus, that staphy prefix means cluster of cells. Okay, so especially the cocci, we see a lot of different arrangements, and when we see a particular arrangement under the microscope, that gives us a hint as to what that identity of that organism is. I realized, I hope a lot of you have checked out those learning objectives that I have posted. I realize that the one that I have posted here is kind of vague. I have written something like, understand the 9 different arrangements, and I don't even think this adds up to 9. That was a carryover from the old textbook. So I want you to know the terminology that I've listed here: the pairs, the tetrads, the Sarsina, chains, and clusters. So just to be a little bit more clear there. The bacilli, we don't see as many different cell arrangements there because they only divide on one plane. Okay, they don't divide long ways, in other words. So sometimes we see some chaining, but typically, or maybe pairs, but typically not much fancier than that. Okay, so just a little terminology there. And then, so starting on the outside of the bacterial cell, the first thing that we come to is the glycocalyx. So this is a term for a slimy substance that most bacteria secrete outside of their cell wall. And it's very common for bacteria cells to secrete this because it has so many advantageous functions to the cell. It's composed most often of polysaccharides. So repeating sugar subunits generally. There is a few exceptions. There are a few organisms that have a little bit higher concentration of protein that makes up their glycocalyx, but most commonly polysaccharides. And what advantages does this bring to the bacterial cell? A big one is attachment, either to other cells in terms of forming some kind of biofilm, or maybe to some host structure. For instance, the bacteria that survive in your mouth live in your mouth because they make some kind of glycocalyx so they can stick to our teeth. Also, another big function is protection from desiccation. Okay, they're going to dry. Bacterial cells, they do not want to dry out. That's usually will lead to their death. So any kind of adaptation they have, keep their moisture in, is going to be helpful. Bacteria that we would maybe cough out into the air, for example, if they land on some surface, if they have a capsule, they're just going to stay viable for a little bit longer. Okay, so help keeps them from drying out. And also protection from phagocytosis by white blood cells. Okay, so a bacteria trying to establish an infection in our body, our white blood cells have a much harder time phagocytizing them if they have a capsule. Now, we have ways to eventually mount a more specific response, but at least with our kind of first line of immune cells, it's harder for them to get rid of bacterial cells with a capsule. Okay, and sorry, I said the term capsule. A lot of times we refer to the glycocalyx as a capsule if it's fairly thick. It's a thick capsule outside of the cell, so you can see in the scanning, transmission electron micrograph. And by the way, they add the color in here. We don't use dyes when we do electron microscopy. They just add that in to try to, highlight different structures. But the orange here is a true capsule, pretty thick on the outside of the cell. If the capsule or the glycocalyx is a little bit more loosely organized, we would probably more refer to it as a slime layer rather than a true capsule. Okay, so it's kind of a continuum there. A lot of organisms, again, at least make some kind of slime layer. It does all the same things that a true capsule would, just not quite as well, basically. So same functions. All right, and then this is just a slide with some images from your textbook, just highlighting, hey, the capsule's on the very outside of the cell. We're going to get to some of these hair-like appendages next, and then into the cell wall structure. When you do a capsule stain, usually your strategy is to stain the background. So you're going to get a chance to do that if you're in lab. It might look something like this. You try to stain the background because if you want to see the capsule, it's very difficult to get dye to stick to the capsule directly because the sugars that make up the capsule don't have much of a charge, and the dyes don't stick to it very well. So we stain the background, and then the unstained part is the capsule itself. So here's a little image that you can see a little bit better. Again, all this unstained part around the cells, that's the capsule. And we need a special staining procedure to do this because when we gram stain, we're always going to be heat fixing the cells, and that heat always destroys the capsule when you gram stain it. So you don't use any heat when you capsule stain, and then you realize how big that capsule often is. All right, some organisms will have flagella, not all of them, but the function of flagella is for motility. So moving towards some nutrient, we would call that chemotaxis, or if it's a photosynthetic organism, you might call it phototaxis if it's moving towards the sunlight, but it's moving towards some attractant, and not all bacteria have it. I will say it seems like the cockeye-shaped organisms, they do not have a capsule. All right, so there may be some exception to that somewhere, but it seems like all the other cell shapes may have capsule, may have a flagella, may not, but it seems like the round cells do not. You don't ever see that. So not all bacteria have them. So if they're motile, they do have flagella. If they're non-motile, then they don't have flagella. And the structure, 3 parts, and the bacterial flagellas are quite different than the eukaryotic flagellas. So we'll get into the difference there. But the prokaryotic ones are going to have a filament, and that's composed of the protein flagellin. So not kind of intuitive there. And then there's a hook region, and that's fastened into the cell wall by the basal body. The bacterial flagellas are not enclosed in a membrane most of the time. There's exceptions to that, but most of the time. So that's why we're doing that in the order that we are, because it's still considered external to the cell wall. All right, so the picture that the OpenStax gave, I didn't like as much as the old one. This is a little bit more complicated than what we really need to see, because we haven't talked about the cell wall yet. So it shows 2 different ways that basal body might look, the part that's attached into the cell wall. But you can see here the filaments. All right, so this comes out this way. And that filament is going to move by rotation. Okay, it spins. And as we'll see, that's different than the movement of the eukaryotic flagella. Okay, so that's going to spin one way or the other. There's this hook region here. And then again, where it's anchored into the cell membrane, that's called the basal body. And don't worry, it's a little bit more, there's a little bit more, rings, because there's more layers of the gram-negative cell wall. Don't worry too much about that. Okay. So some terminology that we use to describe flagellar arrangements, a lot of variety here, whether organisms have a single flagella or many flagella. If they have one, and pay attention to the prefixes here, mono, if they've got one flagella, we call that monotrichous. So that's always going to be a polar flagella, meaning at one end of the other. Okay, so you're not going to have a flagella sticking out like that from the body like that. Okay, amphitrichous. What does that prefix amphi mean? Anybody know? You know what amphibians are? They live in both the water and the land. Okay, so amphi means both. Okay, so a flagella at both ends. Lophotrichous, that prefix lopho means tufts. Okay, so a tuft of flagella, either at one end or the other, usually, occasionally have it at both ends. And peritrichous means you have flagella all over the cell. So perimeter means all around the room. So peritrichous, all around the cell. And this term, what's more common than the other? I don't know. There's a lot of examples of all of them, actually. And that one exception of the flagella that's enclosed in a membrane, we call that the axial filament. And remember, this is the kind of flagella that the spirochetes have. Okay, so any organism that has that morphology, that helical morphology. And so you can see here, down here, how the flagella actually is enclosed in a membrane, and it goes down the whole length of the cell. It's actually that flagella is kind of a blow up here, is composed of many endoflagella. And each one of those endoflagella kind of spins on its own. So that propels that organism in a corkscrew-like manner. So this example here is the organism that causes syphilis, Treponema pallidum. So what kind of motion does this produce? So we talked about how the flagella spin very rapidly. We'll see some, well, I'll try to bring some animations up here. They spin so rapidly, you really can't even see them, and they're so small with light microscopy anyway. But when they move, when the flagella spin in one direction, so when the flagella, if you just have one flagella, it spins one way and the cell goes one direction. And if it spins the other way, the cell goes the other direction. It's pretty simple. But if you have the peritrichous flagella all around the bacteria cell, when they spin clockwise, each flagella kind of spins on its own. So that produces a tumbling effect. When they spin counterclockwise, all the flagella kind of tuft together and work together to produce purposeful movement in one direction. So when a cell has that peritrichous flagella, you get a series of runs and tumbles. Okay, so they move counterclockwise, you're all going together, but the attractant is over here. So it senses it's getting further away. Okay, so then the flagella switch and all move the other direction, which produces some tumbling, and then it goes off in another random direction. And if it's getting closer, it would continue to the attractant, it would continue. In this case, you would have to have a series of runs and tumbles till it would actually go in the direction that it wanted to go. Okay, so we'll see if we can get any of these to work. I think it's just a little bit, it was a little bit slow. Oh, good. Okay, this is a really short one. So this is obviously a cartoon, but if you watch it long enough, you see those are tumbles, runs and tumbles. Okay. Let's see. These are a little bit, like I said, you can spend a lot of time looking for good animations here. This one, I think I wanted to show you the last half here. Let's see. Yeah, so this is what, this doesn't look too exciting. This is probably bright field microscopy, just doing a wet mount, I'm guessing. It could be phase contrast. Remember, with phase contrast, we don't have to stain the cells and we can see them. But if you kind of watch them, there's a few that just sit and spin around. They're probably stuck to the glass, the cover slip. There's some dead ones probably, and then other ones they'll shoot off straight and then they'll tumble around and then they'll go off in another direction. So it's kind of fun to watch them. Again, remember, when we stain cells, that kills them. So then we don't get to watch them. One more, let's see. This one, like I said, you could spend a lot of time doing this. Yeah, same idea there. You get the idea. So they're gonna be kind of fun. Okay, all right, questions about flagella? They're not actually using ATP. I think it would take a lot of energy for the cell to keep that flagella spinning around. They actually use a proton motive force, which we'll talk more about when we get to metabolism, what that proton motive force is. So we won't worry about that right now. Okay, other structures that we see outside of the bacteria cell on some cells. And most commonly on gram-negative cells. I know we haven't talks about what that means, gram-negative exactly. But there may be a few exceptions to gram-positives, but as far as I know, it's all gram-negatives that either have fimbriae or pili. So these are other appendages. They're shorter. Both of them are going to be, well, especially the fimbriae. These are pretty short, and a cell would have many of them. So you can see here it's these kind of short hairline appendages. The long ones there are flagella, and then the shorter ones are the fimbriae. Both fimbriae and pili, I'll go out this way, they both have the same composition. They're both made of a protein called pilin. So again, flagellin for flagella, pilin for pili and fimbriae. Because the fimbriae and pili have the same composition, sometimes those two terms are used kind of interchangeably, which it can be a little bit confusing, but usually we call this structure fimbriae if their function is for attachment. Okay, so if there's lots of them and they're pretty short, they're going to be used for attachment. So for example, E. coli, when it has lots of fimbriae around its cell to attach into the endothelial cells of the large intestine. Okay, without those fimbriae, it would just get washed out of our digestive tract. So those are an essential factor for it establishing an infection in our body. Underline that. Pili, usually a cell will only have one or two, and they're usually a little bit longer if they're considered pili, and their function is for conjugation. And we're going to talk more about what conjugation is later, but that's just a way that cells can share DNA. So the pili is used to pull two cells close to each other so that DNA can be shared through a pore. Okay, the DNA doesn't actually travel through the pili. It's just to bring cells closer together. All right, so now we come to the cell wall. So we're going to spend a lot of time on the cell wall and really look pretty closely at its structure because it's something that we will return to many times throughout the semester. Function of the cell wall, basically it imparts the shape to the cell and it gives the cell some strength in terms of withstanding differences in solute concentration. So like a plant cell, if you get a big influx of water, because a plant cell has a nice sturdy cell wall, it's not going to burst as easily as, say, a red blood cell would if it's in a hypotonic medium. So we'll talk more about osmosis and review that later, but it's really important. Almost all bacterial cells have a cell wall. Like I said, there's exceptions with everything, and there's one genera that doesn't have a cell wall, but mostly they do have a cell wall. It's not the thing that decides what comes in and out of the cell. That's the cell membrane. Okay, so it's mostly strength. It's really porous. As long as something is small enough to fit through it, it's going to go right through without any trouble. Composition. It's varied a little bit depending on the species, but all bacteria in their cell walls will have peptidoglycan. Okay, a layer of peptidoglycan. And again, not archaea with cell walls, not eukarya with cell walls. Okay, so that compound, we're going to mention that a lot, peptidoglycan, that is unique to organisms in that bacteria domain. What is peptidoglycan? Well, the glycan, that should make you think sugar. Okay, so that is composed of two sugars, NAM, which is N-acetylmuramic acid, Again, this is a sugar, very similar in structure to glucose. And the second sugar is NAG, N-acetylglucosamine. Okay, so those two sugars alternating, and they are cross-linked by peptido. That should make you think peptide. So it's cross-linked by amino acids. Okay, so this is a very strong structure. It's kind of like a chain link fence is a good analogy here. So if we look at, well, first of all, if we look at structure of those sugars, don't memorize structures. This is just to show you, hey, here's the structure of glucose. The N-acetylglucosamine is, again, very similar, only different at one of the carbons there, and N-acetylmuramic acid is very similar as well. And those two sugars are very similar to each other. Okay, so similar to glucose. And if this is kind of the best visual that I could find. You have the alternating sugars, and then these connecting here and between layers, connecting amino acids. So there's slight difference in the linkages of the amino acids, and there can be different amino acids there from species to species, but that structure peptidoglycan is very similar. Okay, and we'll finish here with thinking a little bit about how that peptidoglycan is able to grow. It has to do that really carefully, because in order for the cell to expand, right, when a bacterial cell gets a little bit bigger and then it splits into two, okay, so that cell wall has to be able to grow. So new NAG and NAM subunits have to be inserted in there for that cell wall to grow. Okay, so that requires the use of at least two different enzymes. I know there's at least a few more. Autolysins, these are enzymes that cut those glycosidic linkages between NAM and NAG. Okay, so if we think about it like this, there's these linkages, something like that. So the autolysins will cut those glycosidic linkages so that new subunits can be put in, as well as the peptide, the bridges. And then transpeptidases, these are enzymes that are going to reseal the amino acid bonds. There's another enzyme that reseals those glycosidic linkages that we're not going to worry about. Okay, so I'll come back to that animation in a minute. This is the best picture that I could find of this. If this is inside of the cell, right? So those new subunits are going to be manufactured. Those new sugars are manufactured inside of the cell, and then they're brought through the cell membrane through special channels. Don't worry about that, and inserted into the cell wall. Okay, after the autolysins have cut them. Okay, and then the transpeptidases, that's what the TP there, that's resealing up those amino acid bridges that were cut by the autolysins. Okay, and again, this has to be done really carefully, because if those transpeptidases aren't doing its job, then that's going to lose the integrity of the cell wall. You're going to not have those bridges formed where they need to be. And importantly, penicillin binds to transpeptidases and keeps those enzymes from working. So basically what penicillin does is weakens a cell that's growing. It weakens its cell wall so that it's going to burst if it's in a low solute concentration. So that's the importance of that. Okay, so we will continue with the cell wall on Friday. Make sure you're starting that homework if you haven't done that yet.